Okay, hello everyone. Good evening all. Good evening. Good evening, Grishma. Good evening. Okay, what about others? Good evening, Jagannath. Good evening all. Good evening all. Okay, let others also very quickly join. I'll give just 1 minute of time. People are only serious for a few days, huh? Okay, no problem. Okay. Okay, so friends, in the last class, good evening all. We were doing about the photosynthesis topic, and if you quickly recall, my friends, so let's solve this 2022 questions also. In the last class, we were discussing about the light reactions and dark reaction. If all of you quickly recall, we have started with what, friends? We have started with studying the light reactions. And if all of you quickly recall my words, I said that the photosynthetic pigments are broadly categorized into principal pigments, then friends, followed by the accessory pigments. And you can also remember, friends, principal pigments are different types we read, accessory pigments also we read. Later, friends, we started reading about the light harvesting complexes of two type of photosystems. Good evening all, and if everyone quickly recall, we started studying about what, friends? Photophosphorylation. And there are two type of photophosphorylation we studied, my friends. We read cyclic photophosphorylation also, non-cyclic photophosphorylation also. And in just 1 minute I will summarize this. Everyone pay attention. In case of a cyclic photophosphorylation, if you quickly recall the stream of electron transport, I told you, friends, the first primary electron acceptor is what? This is cyclic, don't confuse. Ferredoxin, then followed by plastoquinone A and plastoquinone B. Then, friends, followed by the cytochrome B6F complex. Then, friends, followed by the plastocyanin. Right? Good evening all. This is what we read. And friends, if you all of you quickly look into the non-cyclic photophosphorylation, what did I say to you? P680 from P680 pheophytin, quinone A, quinone B, cytochrome B6F, plastocyanin, P700. And if you quickly recall, friends, from the P700 we studied very clearly that A0, A1, iron sulfur X, iron sulfur A, iron sulfur B, ferredoxin, FNR, NADP plus and NADPH. Till this particular slide we have studied, and you remember I have told one important statement to all of you. In case of a cyclic photophosphorylation, we will only produce the ATP. We are never going to produce the NADPH at all. That is what we studied actually. Now, non-cyclic photophosphorylation, what is the statement you have to remember? In case of a non-cyclic photophosphorylation, friends, we can produce the NADPH also, friends, and we can produce the ATP also. But friends, so far what I told to you, in this particular slide, we have discussed only the production of NADPH. And friends, undoubtedly, in the non-cyclic photophosphorylation, ATP is also going to get synthesized. So first, let's look into the 2022 question once quickly. Okay. I want every one of you to answer this question which was asked in the 2022 paper. Okay. Very simple, very direct question. Okay, wait a minute. First, let me finish this particular ATP production also. Then you can answer the question in a simple and easiest way. Okay, so we read NADPH production, and friends, now we are going to read what? ATP production. And friends, all of you listen. Good evening all, please continue. When I said the synthesis of ATP, every student please remember, this slide is little confusing slide. Just listen to me. How we are going to produce ATP in the non-cyclic photophosphorylation. All of you listen my statement. We are going to produce this by proton motive force. What force, friends? Proton motive force is the one which is involved in the biosynthesis of this particular ATP molecule. Always keep that particular point in your mind without fail. Now everyone pay attention. I will explain the ATP synthesis in a simple and elaborative way. Just listen to me carefully first. Now friends, pay attention. This is the P680 and this is, what do you call, friends? Plastoquinone. You all first listen to me for 10 minutes continuously. From the P680, the electron donated to whom? Pheophytin. I am explaining you in this particular slide how exactly the ATP synthesis occurred actually. From the P680, the electron sent to the pheophytin, and from the pheophytin, where the electron will send to? Electron will send to the plastoquinone. All of you listen my statements carefully. When this is a too confusing concept, I will explain twice. Pay attention. From the pheophytin, electron comes to where, friends? Plastoquinone. Now plastoquinone immediately reduced to what, friends? PQH2. Now all of you has to listen an important statement. If the PQ want to reduce to the PQH2, do you think only electrons are sufficient? Not at all. Anyway, electron is given by the pheophytin, and it is also required what, friends? Proton. Now friends, the question is very clear. Who is going to donate this particular proton? This particular proton coming from the stroma. Very simple, friends. So all of you listen to me carefully. So from the stroma, plastoquinone accept the proton. From the pheophytin, it accept the electron. PQ reduced to PQH2. But you also know very well, PQH2 is not going to keep the electron with itself. PQH2 send the electrons to what? Cytochrome B6F. We all commonly know that. Now listen my statements carefully. Now this particular PQH2, upon sending the electron to the cytochrome B6F, PQH2 oxidized back to the PQ. Now anyway, electron sent to the cytochrome B6F. But my friends, what is the fate of the proton? All of you pay attention. Proton can be released into the lumen. All of you pay attention. At the same time, when the water molecule break down, we will get 2 protons, 2 electrons and half oxygen molecule. That protons are also released into the lumen only. Now the concentration of protons in the lumen, it is getting increased continuously. I'll summarize, my friends. Every one of you pay attention very clearly. In this slide, I am explaining to all of you how the ATP synthesis can occur in the non-cyclic photophosphorylation. As I said, ATP biosynthesis can occur with the help of a proton motive force. Whenever P680 send electrons to pheophytin, from the pheophytin, electron will come to plastoquinone. From the and plastoquinone immediately get reduced to PQH2. But if the plastoquinone want to reduce it to the PQH2, compulsory, what does it require? Compulsory, it required the proton also. Good evening all. So proton and electron, PQ convert into PQH2. Question is very clear. PQH2 is not going to keep the electron with it. So that's why PQH2 is going to send the electron to the cytochrome B6F. Now PQH2 immediately, what it is doing? It is releasing the proton into the lumen. Like that, all the protons are coming into the lumen. Once again, my friends, whenever the water molecule break down, where the protons will be released? Undoubtedly, protons will be released into the lumen only. Now, you only try to understand the concentration of the protons in the lumen will be increasing continuously. My friends, whenever the concentration of protons increasing continuously, what will happen here? All of you pay attention. Now this particular protons starts moving from the lumen. Where? From the lumen into the stroma to maintain the charge balance. We all know very well, we are reading from 10th standard. Proton always move in the form of a wave. You can see here, it is moving like this in the form of a wave. So finally it reached the stroma. Charge balance got maintained. So friends, all of you listen my statements carefully. Proton, whenever it is moving from the lumen to the stroma, how it will move? it will start moving via an enzyme complex. What do you call it, that particular enzyme complex, friends? We will be calling it as the F0 F1 complex. What do you call that enzyme complex? We will be calling it as the F0 F1 complex. Everyone pay attention. This particular proton, whenever it is moving through this particular F0 F1 or ATP synthase enzyme, proton generate the energy. Listen my friends, proton generate the energy. Whenever there is a generation of the generation of the energy, that energy is used by the ATP synthase enzyme to join the ADP with PI. That is how, my friends, we are going to synthesize ATP molecule. Very, 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 very simple point it is. Now, all of you just try to answer this question which was asked in the 2022 June paper. February paper, which was happened in the last month, and which is very easy question. I'll give you exactly 1 minute. I want everyone to answer correctly. Such a easy question, such a direct question, very, 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 very simple question. Just like that we will get 4 marks. No need to study anything. From the photosynthesis only 2 questions came, 1 2 marker and 1 4 marker. You can answer this question without studying. Yes friends, please, please everyone answer. Come on, be active. Saima, what is the answer you have given in the exam? Okay, everyone. D. Runalini, Sunita, Sadguri. Everyone. That's not B, Sunita. Okay. Friends, such a simple question it is. They have asked a very simple and very direct question, that is scheme of electron transport. Electrons from P, look into the option A, every one of you. Fantastic, amazing. The answer is absolutely D. Just like that you can answer, right? The electron from P680 moves from first to quinone and then pheophytin, which is wrong statement. From the pheophytin, it will move to the plastoquinone. So the option A is wrong. Like that only you can answer the question dead easily. Now option B, P700 can receive the electron from what? Plastocyanin. Of course true. Plastocyanin is the electron donor for the P700. And we read very clearly during the light reactions, we will produce the NADPH, no doubt about it. And just now we read where this particular hydrogen ions get accumulated. I have shown you in the thylakoid lumen. Very, very simple. The answer will be D. Now see here, from photosynthesis in your 2022 paper, 4 marker question was came. Only 1 question came. And after some time I will make you solve another 2 marker question also, which come from the photosynthesis. 6 marks are very direct marks and very easy, very straightforward question. That's really great, friends. Let's continue. Now all of you listen. Friends, that's good, fantastic. Everyone has given the correct answer. Now friends, understand what are the major differences between the cyclic and non-cyclic. Is there any necessity to remember the differences? It is compulsory. You have to remember the differences undoubtedly, friends. So all of you pay attention. In case of a cyclic photophosphorylation, friends, there is only one photosystem is involved. Correct na? So it may be anything. Sometimes it may be PS2 also. Sometimes, good evening Sumaya. Please watch the recorded, whichever you missed. We solved just now. We solved 2022 question. Only one photosystem is involved, but here in the non-cyclic, both the photosystem 1 and photosystem 2 both are involved. And very important, friends, in the cyclic photophosphorylation, photolysis of water or the breakdown of water never ever takes place. In the non-cyclic, compulsory breakdown of the water will takes place. That is the reason in the cyclic we can only produce the ATP. We never produce the NADPH. We commonly know that. But in case of a non-cyclic, we know very well we will produce the ATP also, NADPH also. Friends, you only try to understand, since the water molecule is breaking in the cyclic photophosphorylation, friends, of course water molecule is not breaking. Undoubtedly, my friends, there will be a no release of the oxygen also. Very simple point. So oxygen is not evolved as a end product or the byproduct. So friends, who knows, in the next term we can get the questions from this particular statements. Oxygen is evolved as a byproduct because water molecule break down. Since the water molecule is breaking down, undoubtedly oxygen can be evolved as a byproduct. And my friends, it is a very important, it is necessary, we have to remember the inhibitor. CSIR, in CSIR exam, my friends, it is a very important, you people has to remember compulsory what you have to remember, inhibitor. Not only unit 6 or unit 1 or unit 2, all the 13 units wherever you read the inhibitors, regulators, stimulators, all you have to remember. Anyway, let me explain you. See friends, for the for photosynthesis, PPT will be 100% sufficient. The question won't go beyond this level. Okay, now listen to me carefully. What are the various type of herbicides that block the electron flow? So in this here I have mentioned 3. One we call DCMU. DCMU, friends, sometimes we will also call it as a diuron, dichlorophenyl dimethylurea. Next one, triazine. Next one, paraquat. What are the differences? Now you only try to understand. It is very important, direct questions you will get. Look into this. This is the P680, right? Photosystem 2. This is what? Photosystem 1. Now you commonly, just now we read in the question also, from the P680 electron comes to PO510. PO510 to what? Quinone A, then quinone A to quinone B. You know friends, what is the speciality of this particular DCMU or diuron? It block. What it is going to block? It is going to block the electron flow between the quinone A to quinone B. In a simple line, I can also tell you friends, DCMU is the one which will block. What it is blocking? It is blocking the electron flow between the P680 and P700. Do not forget this particular statement under any circumstances. Now if you look into the paraquat, I can tell you very clearly, listen my statement. Paraquat basically block the electron flow which are involved in the production of NADPH. Once again listen, paraquat block the electron transport which are involved in the production of NADPH. DCMU can block the electron flow between P680 to P700. If you ask me, sir, very specifically where it is going to block, it is going to block between the quinone A to the quinone B. Never ever forget, because all the time inhibitor questions are important extremely for the part C only, not for even part B also. Okay, everyone answer the question. Question number 76, which is a part C question asked in the exam. Think and answer, don't be hurry. You can answer all the questions very easily actually. Everyone. Anyone? It's not difficult question, simple, direct 4-marker question. You have to choose the right option. So fast, every one of you. Okay. But while choosing the answer, make sure that you are choosing the correct answer. See, simple question for the 4 mark. Absolutely true. Even you no need to look into the another option also, because I was very clearly told you that PS2 to PS1, the electrons are blocked by the DCMU. When the PS1 is involved in the production of NADPH, here the electron transport can be blocked by what, friends? Paraquat. Please kindly do not forget this, friends. And once again, before saying the lesson, I am saying you, I will cover 100% of the concept, which means the question won't go beyond that level. So read the concept very clearly, whichever I am teaching to all of you. That's good. That's good. So friends, finally we understand what, friends? Light reactions of the photosynthesis. Okay, now everyone answered this particular question, friends. Oh, no, not one. 1 minute. Let me teach this, then only you can understand this. Okay, so friends, this is what light reactions. Friends, all of you listen to me. Before going to the dark reactions, we have to read very clearly, friends, 2 concepts. What are the 2 concepts we have to read? We have to read the photoprotective mechanism, and we have to read the most important, physical separation of the 2 photosystem. Friends, studying the physical separation of the 2 photosystems are extremely important for the exam. Very important, because many times the question was asked. Listen to me. Look into this beautiful diagram. You can understand very clearly. Now you can ask me. First, let me ask you one question. My question is, friends, why electrons cannot transport or cannot transfer directly between the PS to PS1? Why? Question is very clear. Why not the electrons can be transported between PS2 to PS1 directly? Why the electrons are basically transported with the help of different electron acceptor molecule? What is the reason? Friends, an example, if the electrons are directly transported between the PS2 to PS1, there will be no way we cannot produce the ATP also, friends. We cannot produce the NADPH also, friends. Always keep this particular point in your mind. Now, listen. So, because all of you listen the statement whichever coming from my mouth. The statement is that in order to prevent, what to prevent? In order to prevent the direct electron flow from photosystem 2 to the photosystem 1, what we have to do? We have to stop the transfer of electrons directly. For that, we have to separate the PS2 and PS1. So how does the PS1, PS2 can be separated physically? Every one of you pay attention. Look into the diagram. This is the stroma. This is what, my friends? Thylakoid membrane. I hope all of you listen, my friends. Thylakoid membrane, what do you call it? It as lamellae. And you can look into the inner space. I told you, inner space we call lumen. Whatever the organelle it may be, I told to all of you very clearly, the inner space we generally call it as the lumen only. Now all of you look into this. This is the light harvesting complex. The light green color one is the, dark green color one is the PS2. Blue color one is the cytochrome B6F, light green color one is the PS1, and friends, this is what F0 F1 complex or the ATP synthase enzyme. Now I want every student pay attention to the diagram. Here you are able to see, right, LHCs. Friends, all of you listen. If you look into this photosystem 2, where the photosystem 2 is located, photosystem 2 located in the tightly oppressed thylakoids. Here, what are these LHCs? Basically, they are thylakoids only, right? Where the photosystem is present, they are present wherever the thylakoids are very tightly attached. There PS2 is located. You can see, my friends, very clearly. Wherever the PS2 is present, it is embedded in the thylakoid membrane very tightly. That's why we can say PS2 is located predominantly in the staked regions of the thylakoid membrane. But friends, if you look into the PS1 and ATP synthase, where they are found, you only look into the picture. You tell me friends, do you think here thylakoids are tightly attached? Not at all. Thylakoids are freely arranged. What do you call it? That as unstaked regions. Or our friends, we will be calling it as the non-oppressed thylakoids. The PS1 and ATP synthase enzyme, they will be arranged in an area where there will be no tight thylakoids are present. Non-oppressed thylakoids are unstaked regions, means which are freely arranged. You can see PS1 is very freely arranged, photosystem 1 also very freely arranged, ATP synthase, photosystem 1, ATP synthase, photosystem 1. They are very freely arranged. Now once again I am reminding you the statement, look into this. If you look into the photosystem 2, photosystem 2 always associated with a very tightly oppressed thylakoids. And friends, if you look into the photosystem 1 and ATP synthase, what is the statement you people has to remember? They are found in the unstaked region. We can also call it as non-oppressed thylakoids which are protruding into the stroma. But all of you look into this particular cytochrome B6F. Friends, don't confuse. Cytochrome B6, everyone listen. Cytochrome B6F is present in the non-oppressed thylakoids also. There is no doubt about it. And cytochrome B6F, it is also present. Where the cytochrome B6F also present? On the non-oppressed thylakoids also. Listen to me. Cytochrome B6F is present in the tightly oppressed thylakoids also, non-oppressed thylakoids also. So what we can say? We can use a word that because it is present everywhere, then I can conclude a statement that cytochrome B6F is evenly distributed throughout the thylakoid membrane and the lumen. Never forget the statement at all. Now everyone answer this question, which is the 4 marker part C question asked in the exam. You no need to read also. You just listen the lesson and answer. That is what I need actually. We have to put less effort in the preparation. We have to work more smartly. Everyone. It's an easy question. Part C. Think and answer. Don't confuse. Take time. Take time. Every one of you. Oh, that's great. Okay. Such a simple question, my friends. Photosystem 2 is located predominantly in the staked regions. What do you mean by that staked regions? I told you, tightly oppressed thylakoids, where the thylakoids are arranged very tightly. And if you look into the photosystem 1, I said to you very clearly, photosystem 1 is found in the unstaked region, which are freely arranged, protruding into the stroma. Absolutely true. Now, cytochrome B6F, they said that it is confined to stroma. This is wrong statement. You can see the diagram. Cytochrome B6F is present everywhere. So that's why we can say cytochrome B6F is evenly distributed. Do not forget that. So the option C is wrong. ATP synthase enzyme is located on the unstaked. So C is wrong, A is right, B is right, D is right. Is it? Okay. So what will be the answer? The answer will be 2. Okay. Okay, Sahani, I will change by the camera by tomorrow class, dear. Now in the middle I cannot change that. I will change it tomorrow. Okay, don't worry at all. Good, 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 perfect. Everyone answer this question. Once again, part C question, which was asked in the exam. I'll change it tomorrow. Don't worry, please. Everyone, think and answer. Correct, Beauty Kumari. So we solved that question, dear. Please move on to the next question. People are giving answers very slowly, friends. You have to be very faster. So fast people are giving the answers. I hope you are analyzing it correctly, friends. Look into this, Sumaya, slowly, dear. Every one of you, please look into that. Others. 2. How's 2, dear? My friends, just like this, you can answer this particular question. You only look into the option P correctly. They said that the PS1 reaction center and PS2 reaction centers are uniformly distributed. Why they both are uniformly distributed in the both granular? I think everyone given wrong answer. I don't know why. Just now I have explained you very clearly, no friends. How the P will be correct? Tell me first of all. They said that PS1 reaction center and PS2 are uniformly distributed in the granule and stromal. Absolutely wrong, right? Which is uniformly distributed? Only the cytochrome B6F. This is the only one which is uniformly distributed. Then option P is absolutely wrong statement. The electron donor for the P700 is the plastocyanin. You only try to look into this. Let me tell you. This is the question asked. See, I want you to know one interesting point here. This is the question. I'm just giving a hint to you. This is the statement and this is the question asked in the 2017. If you look into the 2020-22 June question, February question paper, whatever we have solved, you only look into this, the same statement given. P700 can receive the electron from plastocyanin. There will be no change in the statement also. The electron donor for the P700 of PS1 is plastocyanin, and from the P700 it is electron is accepted by A0. Obviously Q is right. Anyway, once you identify P is wrong, you can eliminate option A, B, C, D will be the correct. The core antenna of P700 are bound to key proteins PSA and PSB. Absolutely right. Cyclic electron flow occur from the reducing side of PS1 and B6F complex. We read in the differences. Just now I shown you, right, differences. We read over there in the cyclic electron flow, we will only produce the ATP. We never ever produce the NADP plus. Very, very, very simple. The correct answer will be, friends, D. See, friends, questions are very easy. Okay, listen the lessons carefully and you can answer. See, when it comes to the plant physiology, when it comes to the, that is unit 6, unit 4, unit 3, unit 7 and unit 2. If you read my lessons correctly, you can answer. Listen my question carefully. Listen my statement. If you listen my lessons very carefully, you can answer any kind of a question from this chapters without reading even. But when it comes to the unit 1, unit 5, unit 10, unit 11, unit 12, unit 13 and followed by unit 8, you have, unit 9 genetics. Here you have to listen also, you have to read also. But here without without reading also, by listening the lesson 100%, you can answer the questions. So that's why pay attention. That is good, friends. So perfectly what we have understood right now, the light reactions. So friends, before going to the dark reactions, there is a last concept is left in the light reactions. We will be calling it as the photoprotective mechanism. Friends, it will be little confused concept. I am requesting all of you, give me 10 minutes. I will explain step by step briefly from basic to the advanced level. Just listen to me first of all carefully. Now, what are this particular photoprotective mechanism? You only try to understand. Let's take an example of human being. Whenever we are exposing to the high intensity of sunlight, what will happen? We will easily try to escape. Yeah, correct. You have done. Okay. Yeah. You have done wrong by eliminating the option P. No problem, dear. Upon practice, it will come. That's why you have to eliminate the question slowly. Don't be hurry. Peace of mind is very important. Yeah, what I am saying, suppose example, we are human being. Whenever we expose it to the high intensity of sunlight, obviously we will try to escape, because we can move easily, we can flee easily. But you only tell me, do you think plant can move? Plant cannot move. So whenever there is a high intensity of sunlight, plant has to protect themselves compulsory. Now, how the plant is going to protect themselves? First of all, listen my statements carefully. What happens when the plant start exposing high intensity of sunlight? What happens when the plant start exposing to the high intensity of sunlight? First of all, listen. If you ask me a question, please listen my statements which I am using all the time. What is the function of a chlorophyll? The function of chlorophyll is what, friends? Conversion of a light energy to the chemical energy. That is the simple function of a chlorophyll, and this particular chemical energy only we will use it to fix the carbon into glucose in the dark reaction. We all commonly know that. Now my question is very clear to all of you. Do you think the chlorophyll molecule can absorb the entire sunlight? That is not possible at all. Listen my statements carefully. Imagine I am a chlorophyll molecule. I am a chlorophyll. Whenever I am absorbing the light, what I will do? How much amount of the light I can absorb? Only that much amount of the light I will absorb, and I will convert that much light energy only into chemical energy. If the light is coming from the sun continuously, do you think I will absorb everything and I can convert into chemical energy? I cannot do that. So I am a chlorophyll molecule. Being a chlorophyll molecule, I will absorb limited amount of the light energy and convert into chemical energy. But what is the condition? The condition is there is a high intensity of sunlight. The sunlight is coming continuously. Now what is going to happen? Everyone pay attention. Now this is one chlorophyll, this is another chlorophyll, this is another chlorophyll. You know, friends, what is happening? This chlorophyll also absorbing the highest sunlight, this also absorbing highest sunlight, this also absorbing highest sunlight. These three are unable to convert the light energy into chemical energy. And because of the continuous absorption of the light, what is happening? They are getting excited. What happened? Three chlorophyll molecules get excited. Listen to me, friends, please. Three chlorophyll molecules get excited. They can form a structure. What do you call it? That particular structure, friends? That particular structure only we can call it a triplet. Now, all the student has to remember one important point. When the chlorophyll molecules are in the triplet, always remember they are having a long lifespan. Very long time they can survive. Now you can ask me a question, sir, what is the function of this particular triplet? I want every student has to understand this triplet basically go and activate several free radicals. It activate what, my friends? Free radicals. What are the example of free radicals? Singlet oxygen, superoxide and peroxide. What they will do, these free radicals? All of you listen. This all free radicals will inhibit entire photosynthetic machinery, which means it will destroy all the photosystem, destroy all the electron acceptors present on the thylakoid membrane. That process only, my friends, we will be calling it as a photoinhibition. For all of you, I am saying the concept once again. Listen to me carefully. Whenever chlorophyll molecules absorbing the light continuously, what is happening is three chlorophyll molecules absorb the light continuously. three chlorophyll molecule get excited, excited chlorophyll molecule, they can form a triplet structure, and this triplet structure having the longer lifespan, this triplet immediately going to activate several free radicals. Whenever they activate several free radicals, what is going to happen? There will be a, once these free radicals get activated, they will destroy all the photosynthetic machinery present on the thylakoid membrane. Hence, photosynthesis will stop. What do you call this particular process? We will be calling this as a photoinhibition. Now everyone answer my question. How the plant is going to avoid this particular photoinhibition? How the plant is going to prevent this particular photoinhibition? By an important mechanisms, we will be calling it as, friends, photoprotective mechanisms. I will be explaining the mechanism very clearly. Just listen to me carefully. I will explain twice. I want you to just pay attention. Actually, this is the diagram present in the Lincoln and Tays book. Okay, and that time when they have asked a question in the 2018 or 17, I feel they have given the direct diagram. Whatever the diagram they have present in the Tays book, and they have asked the question. Anyway, look into the question. I am explaining to all of you how the plant body, by using the photoprotective mechanism, they are going to inhibit the process of photoinhibition. I'll explain twice, just pay attention. Now what is happening? See here, first, suppose example, all of you listen. From the sun, how many photons are coming continuously? I'm just giving an example. From the sun, 100 photons are coming continuously. But now you only tell me, I have clearly told to you, for the chlorophyll molecule cannot absorb that much. How much amount required for the photosynthesis? They will use only that much. Listen, an example I am telling to all of you. Out of 100 photons, just listen my friends, only 50 photons are required for the photosynthesis. Then only that many photons can be used in the form of a photosynthesis. Now how many photons are left? 50 photons are left. What did I tell to all of you, my friends? If this particular 50 photons are not utilized or not released, what happen? They can form the triplet. Triplet activate the free radicals. Free radical will inhibit the photosynthetic machinery, which leads to the photoinhibition. Now, how the plant is going to release this? Listen to me. There are many kind of a defense mechanisms are there. The first line defense mechanism, all of you listen my statements carefully, please. The first mechanism, the first photoprotective mechanism, we called photochemical quenching. I'll explain twice, listen. Photochemical quenching. What do you mean by the word photochemical quenching? Releasing the photons in the form of a heat. That is called quenching, releasing in the form of a heat. That is the first line defense mechanism. Now you will get a doubt. Sir, all the 50 photons are releasing really? No, I can say, my friends, hardly 20 photons only released. But still how many photons are left, my friends? 30 photons are left. Now you know, friends, what the 30 photons will do? They will form the triplet. Now what the triplet is going to do? The triplet activate what, friends? Several free radicals. All of you pay attention. What are the particular free radicals? Superoxide, singlet oxygen, hydrogen peroxide, hydroxyl radical. Now if we don't inhibit all this free radical, they will immediately block the photosynthesis. Immediately which defense mechanism will be active now? Second line defense mechanism will be active. What are the examples of the second line defense mechanism? Carotenoid, superoxide dismutase, we can call it SOD, ascorbate, SOD, ascorbate, and carotenoids. These three we call it as a secondary defense mechanism. They are actually involved in destroying this particular free radicals. So out of 30, let's take an example, 20 photons we have destroyed. then still 10 photons are left, right? All of you, every student has to remember what this particular 10 photons will do finally. They will go and damage a protein present on the PS2. What is the name of that particular protein present on the PS2? We called D1 protein. All of you has to remember, sir, what is the function of D1 present in the PS2? You all know, right? Whenever a water molecule break down, we read, right? There is a release of 2 protons, there will be a release of 2 electrons, and there will be a release of half oxygen. So how the electrons can be received by the PS2? The electrons received by the PS2 from the D1 only. Now that excess photon will come and inhibit the D1. Then do you think the photosynthesis will occur? Never. So once there is a damage to the D1 of PS2 occur, you know what the plant will do, friends? Immediately plant will start the denivosynthesis process. They will do the process of denivosynthesis and immediately, friends, they will synthesize the new D1. By that they can inhibit the process of photoinhibition. I will summarize for all of you, friends. Just pay attention. Question is very clear. How the plant is going to inhibit the process of photoinhibition? What are the various type of photoprotective mechanism? Here I told you 100 photons, 50 photons, 30 photons, just to make you understand, that's all. So from the sun 100 photons are coming, but for the process of photosynthesis, chlorophyll molecules are just required 50 photons only. So 50 photons are used for the process of photosynthesis. Now what is happening? Out of 50 photons, 50 are left, right? Among these 50, nearly, friends, 20 photons we will be released by using the first line defense mechanism in the plant body. What is that first line defense mechanism? We will be calling it as a releasing in the form of a heat, widely called as what, my friends? Photochemical quenching. I am requesting all of you, don't forget the statements at all. Photochemical quenching. And what will be the second line defense mechanisms? So here 20 photons are over, 30 photons are left. What the 30 photons will do? They will activate the triplet state of chlorophyll. They will activate all the free radicals. Now we have to inhibit these free radicals. How can we inhibit the free radicals? By using carotenoid, superoxide dismutase, ascorbate. What are these? Second line defense mechanism. Even little photons. Say an example. 10 photons escaped the second line defense mechanism also, friends. They finally attacked and damaged the D1 of the PS2. As a result, what happened? Photoinhibition occur. So immediately plant will start denivosynthesis. They will synthesize the new D1 protein and then it will inhibit the process of photoinhibition. These are all the photoprotective mechanisms present in the plant. Now all of you pay attention and try to answer the question very clearly. This is the 4 marker. Part C, direct question. This is asked once. Next time they just given the diagram. Twice they have asked the question. All of you. Simple question, friends. Slowly, friends, eliminate the options carefully. In this, it is an easy question actually. Very simple question. It's a part C. So fast. Good. Please reanalyze, analyze. Fantastic. Can anyone tell me the answer? Friends, the question is very simple, right? Whichever I have explained you in the diagram, that is only question. Obviously, what will be the answer? The answer will be D. Correct, na? So the question says that The photon intensity captured by the green plants is used in the following process: photosynthesis, generation of heat, activating the free radicals, damage to the D1 of protein PS2. They have asked a question, when the photoinhibition occur. I told you very clearly, right? If we can't repair the D1 protein, if we can't synthesize the new D1 protein, obviously what is going to happen, friends? Which leads to the inhibition. No, Sahani. It's not C, dear. The question says that when the photoinhibition occur, photoinhibition finally, first mechanism is what? Heating. We will release something. Second one is what? Second line defense mechanism. Third one is what? Finally D1 protein. If we don't synthesize the new D1 protein, once it gets damaged, then what will happen? That leads to the photoinhibition. That is what I told you in the diagram, right? You only look into this. I have told you very clearly, oxidized D1, if we don't synthesize the new D1, then only photoinhibition occur. You are getting my point? Then how the C will be the answer, friends? C cannot be the answer. Excess photon is used for the generation and formation of toxic product. You only tell me, but what is the question asked? The question asked is when the photoinhibition occur. They did not ask whether the photon intensity produce the toxic products or not. That is not the question. When the photoinhibition will happen? Photoinhibition will happen when the D1 protein get damaged. Very simple, Deepika. There are 3 chlorophyll molecules are there now, dear. Because of the triplet structure, the protein synthesis will occur continuously. Protein synthesis will occur continuously. The RNA molecules also synthesized continuously. You know, right? Why the chlorophyll get degraded faster, Deepika? Chlorophyll get degraded faster because of the RNA and proteins only. If RNA and proteins will degraded, chlorophyll will turns into yellow. The leaf turns into yellow. But here, chlorophyll molecule, they continuously synthesize proteins and RNAs which required for the long lasting. By that, they can survive for long time. Friends, I hope every one of you clear with the light reactions. We did not miss even a single point also. If we get a question from here, there is a no doubt. Undoubtedly, we will get the question from the area, whatever we studied. So you use the PPT 100% and I will only provide you the questions for this entire plant physiology unit 6. Once the chapter get over, practice those questions without fail. Okay, question won't go beyond that level. So next one is what, friends? Dark reaction. If you quickly recall, our ultimate goal of the photosynthesis is what? Conversion of the carbon dioxide into glucose. This is the ultimate goal of our plant, but of our photosynthesis. But what we read, plant can't fix the carbon directly into sugar. So for that, what do we require? We require ATP, we require the NADPH. That is what we have done. First we will synthesize the ATP and NADPH. Then immediately what happen? By using this ATP and NADPH, plant is going to reduce the carbon into glucose. That's a simplified point. Now we already produced the energy. By using that energy, plant fix the carbon into glucose. Now you only understand, if I am a plant, if I want to fix the carbon into glucose, I do not require light. What I require only energy. I don't require light. So that's why we call dark reaction. Dark reactions doesn't mean that they are occurring in the dark, which means that they do not require light. We call light independent reactions. And what we are fixing into glucose? We are fixing the carbon into glucose. That's why we can call it as carbon fixation reaction. And we are assimilating, I am a plant, I am assimilating the carbon in my body. That's why I will be calling it as a carbon assimilation reactions also. So friends, based on the dark reactions, based on the carbon fixation, all the plants present on this planet Earth, they are broadly categorized into 3 types. C3 plants, C4 plants, CAM plants. Based on what? Based on carbon fixation. I can tell you one interesting point. 95 to 97.5% of the plants present on the planet Earth, they will follow C3 pathway. C4, what is the interesting point? Especially the plants which are living in the high light intensities. I will tell you anyway. I can tell you one good line here. More than 18,000 types of angiosperms. More than 18,000 of angiosperms will follow the C4 pathway. Camp plants, of course, the desert plants. Simple, right? Okay. So friends, we will discuss the complete dark reaction in the day after tomorrow class. By that the photosynthesis will be completed. And we got one more question from the dark reaction, that is for 2 marker. Very easy, and just like this we can answer that particular question even. So friends, once again I am just telling to all of you, for unit 3, unit 6, unit 4, unit 2, you strictly depending on my PPT 100% and just read Pathfinder just once, that's all. Pathfinder once and practice the questions which I will provide to you. That is sufficient. Remaining chapters I will tell you how much PPT is useful, what you have to read extra. My PPT will be useful for you 90%, but for the 10% I will tell you what to do. Okay. Okay friends, that's all for now. But I am giving you word, whatever the questions that are present on the 2019, we will solve without looking the papers. I mean say without looking books. Just listen the lesson and we will solve the questions. Easy. Okay friends, that's all for now. Thank you so much. I appreciate all your patience, and I am requesting all of you once again, please kindly forward the link to many students. By that everyone can attend. Please forward the Telegram links also. Okay. Thank you friends. Thank you so much. That's all for now. Please give your comments in the chat box. Thank you all. I'll see you in the tomorrow class 8:30 p.m. sharp.